So good afternoon, everybody. So I will talk today about the role of the gut microbiota in liver diseases, and I will particularly focus on the way we can use germ-free animal models to prove the contribution of the gut microbiota to these pathologies. I will talk about two different liver diseases, the non-alcoholic liver disease, fatty liver disease, NAFLD, and the alcoholic liver disease. So the NAFLD is a pathology covering different stages with different degrees of severity. The first stage is a steatosis, which corresponds to an accumulation of triglyceride in the hepatocyte, so the fatty liver. It's present in almost all obese people, but also 20% of non-obese people have a steatosis. And in 20 to 40% of patients with steatosis, an inflammation occurs leading to the NASH, non-alcoholic steatohepatitis, which is associated with insulin resistance and which can lead to fibrosis, cirrhosis, liver cancer, and to death in 10% of the cases. And of course, as the incidence of obesity increases, the incidence of NAFLD increases as well. The alcoholic liver disease covers more or less the same stages. So a long-term heavy drinking leads to a steatosis. It is reversible after a few weeks without alcohol consumption. And some of the heavy alcohol drinkers stay at this stage all lifelong. But a fraction of alcoholic people develop an inflammation, so an alcoholic steatohepatitis or fibrosis or cirrhosis, and all these severe stages of the pathology, which can be very severe. And this severe form have a mortality rate, for example, at almost 50% at 1 month. But as I told you, for the obese and the alcoholics, so some of them stay at the steatosis stage. And so the question is that why only a fraction of obese people and heavy alcohol drinkers develop the severe stages of the liver diseases? And our hypothesis is that the gut microbiota may be one of the factors explaining why a fraction develops the severe stages. Where does this hypothesis come from? As Joel said, a dysbiosis, so an imbalanced microbiota, has been associated to different pathologies, so you can see a lot of them here, including the liver diseases. But as Joel told also, the dysbiosis does not tell about the causality. So how can we prove the causality between the gut microbiota and the disease? I believe that germ-free animal models and gut microbiota transplant, Neve gave a lot of good examples, can be a very good way to prove the causality. So what you can see here is a letter from Louis Pasteur, published in 1885. And in this letter, in French, Louis Pasteur wrote, "If I had the time, I would try to feed animals from the birth with completely sterile food," so with the idea to create germ-free animals. And he said, "And I have the preconceived idea that in this condition, life would become impossible." So Louis Pasteur already thought that bacteria was important for life, or maybe necessary. He added, "If I could develop this system, we could try to study the digestion by adding single of a mix of microbes. And finally, whether life would be impossible or in contrary easier, it would be of great interest to do the experiment." So 132 years ago, I think, Louis Pasteur understood that the gut microbiota is important, and probably that germ-free animal models would be a unique tool to study the role of the gut microbiota in physiology. So a few decades later, his idea became true, and you can see two photographs from our germ-free facility in our Michaelis Institute in Jouy-en-Josas. And so in the rest of my presentation, I will present you how we can use these germ-free animal models to try to demonstrate the causality or the contribution of the microbiota in liver diseases. Okay, but gut microbiota is essential, but one can live without bacteria, or to be more accurate, mice and rats, I don't know for us, but mice and rats can live without bacteria. So can we prove that the gut microbiota play a causal role by transmitting the different susceptibility to this liver pathology via microbiota transplant to germ-free mice? So the first study is about the non-alcoholic fatty liver disease. It originates from this observation. Oh, something got wrong. When you give a high-fat diet to conventional mice, you get heterogeneous responses. I mean that some mice become obese, some others do not, some develop insulin resistance, some others do not, steatosis, exactly. It's the same for all the parameters. And our hypothesis is that the difference in gut microbiota composition are responsible for this heterogeneity of response to the high-fat diet. And so our strategy was to transplant the gut microbiota from 2 mice with the opposite response to a high-fat diet to 2 groups of germ-free mice to see whether we can transmit the different susceptibility or protection to a diet-induced non-alcoholic fatty liver disease by transplanting different gut microbiota. So in the first part of the study, the experimental design was quite simple. We used 24 conventional mice and we gave them a high-fat diet for 16 weeks. And at the end of these 16 weeks, we evaluated the development of the pathology by analyzing the metabolic status, the hepatic lipid metabolism, and the liver inflammation. So I don't want to show you all the parameters that we obtained, so I just show you this slide to explain how we selected our 2 microbiota donor mice. So, I don't know, we don't have a... This is one mouse, the second one, the third one, etc. And the mice are ranked by body weight. And I show you just 2 parameters, HOMA index for insulin resistance and the plasmatic concentration of MCP1, which is a marker of the systemic inflammation. But we had something like 20 more parameters that we have measured in these mice. And so from all these parameters, we selected these 2 mice. One mouse that we call the responder mouse, which developed a high level of insulin resistance, of liver inflammation, systemic inflammation, etc. And the non-responder mouse, which did not develop any of all these metabolic disorders. And as you can see, these 2 mice were the same weight, highlighting that the level of obesity is not correlated to the development of the metabolic, or to the level of the metabolic disorders. Then the next step was to transplant the gut microbiota from these 2 mice, so the non-responder and the responder one, to 2 groups of germ-free mice to see whether we were able to transmit the different susceptibility to non-alcoholic fatty liver disease. So we got these 2 recipients groups, a non-responder recipient and the responder recipient, and we gave them the same high-fat diet for 16 weeks. And now I show you the result that we obtained with these 2 groups of mice. So in the non-responder in blue and the responder in yellow. So this is the weight curve. So they are quite similar, but it was not a surprise. The 2 donor mice were the same weight. But when we looked at the glycemia, we found that only the responder recipient, so the germ-free mice associated with the gut microbiota of the responder mouse, only these mice develop a hyperglycemia and hyperinsulinemia. And so the HOMA index, which is correspond to the level of insulin resistance, was found 2.3-fold higher in the responder recipient compared to the non-responder recipient. When we look at the liver, you can see that only the responder recipient developed a massive steatosis, which was confirmed by a blinded histological analysis and by the assay of the triglyceride in the liver. And using qPCR, we were able to find that only the genes involved in de novo lipogenesis were overexpressed in the liver of the responder recipient compared to the non-responder recipient. So we also analyzed the gut microbiota of these 2 groups of mice, but I don't talk about it today because I thought I had only 10 minutes, so I tried to go fast. So now let's switch to the second study, which is about the alcoholic liver disease. So the objectives of this study was to characterize the gut microbiota composition of chronic alcoholic patients at different stages of the alcoholic liver disease, and to see if we can transmit the different susceptibility to alcoholic liver disease via human microbiota transplant to germ-free mice. So in this study, we included patients drinking more than 80 grams of alcohol per day. You can make the calculation at home and you will see it's a lot. With no antibiotic therapy in the last 3 months, and we assessed the liver injury by a liver biopsy. So the severity of the pathology was characterized by the anatomopathology score of liver lesion and by the number of polymorphonuclear cell infiltrates, so the liver inflammation in the liver biopsy. And from this characterization, the 37 patients that were included were divided into 3 groups. A group with a severe alcoholic hepatitis, which is a very severe stage. At this stage, half of the patients dies in the following months. The non-severe alcoholic hepatitis, they have an inflammation of the liver, but it's less severe, and a group of patients with no hepatitis. So again, all the patients were alcoholics. So just a few characteristics of this alcoholic patient. I won't go to all the parameters, but I just want to focus on 3 of them. The first one is the alcohol consumption. And as you can see, the group with the more severe disease drink less than the other group on average. So highlighting the fact that the severity of the pathology is not correlated to the quantity of alcohol consumed. But if you look at bilirubin and Madurai discriminant function, which are two markers of the liver severity, you can see that they were much higher in the group with the severe alcoholic hepatitis. So we analyze and compare the gut microbiota of these 3 groups of patients, and I will show you the results only for the genera that were significantly different. And as you can see, specifically in the group of patients with the severe alcoholic hepatitis, we found more enterobacteria, more bifidobacteria, more streptococcus, and less atopobium. And again, it's not all the patients were alcoholic, so it's not the effect of alcohol. So the alcoholic patient with the severe alcoholic hepatitis have a profile of gut microbiota different from the other alcoholic patients. But at this stage, it does not tell about the causality. So the next step was to transplant the gut microbiota from this patient with a different degree of severity to the germ-free mice. So we selected 1 patient with no hepatitis and 1 patient with a severe alcoholic hepatitis, which have a profile of gut microbiota representative of their group. And we transplanted their gut microbiota to 2 groups of germ-free mice, and we alcoholized. We gave alcohol, an alcoholic diet to the recipient mice for 5 weeks. It's only 3% of alcohol, but it was a liquid diet, so they had nothing else. So all day or night, because they eat at night, they ate something. It was like a pasta with 3% of ethanol. So it's enough to make some liver diseases. So we quantified the liver inflammation using flow cytometry, and as you can see, in the liver, so in red, this is the mice associated with the patient with severe hepatitis, and in green, the mice associated with the microbiota of the patient with no hepatitis. So we found much more inflammatory cells in the liver of the mice with the bad microbiota, and it was not specific for type of inflammatory cells, so we had more CD3, CD4, CD8, CD19, and natural killer mice cells. I don't know if you are familiar with that. Maybe it's not clear on the image, but when an anatomopathologist analyzed the liver histology of this 2 groups of mice, he found a liver necrosis score 5-fold higher in the mice associated with the bad microbiota. And in humans, usually the alcoholic liver disease does not touch only the liver, but also involve the gut inflammation and others. So we look at other organs, including the gut, and we found an increased gut permeability in the mice with the bad microbiota. I don't go very long into the image, but we found more translocating bacteria in this group of mice compared to the other. And we also found more translocating bacteria in the mesenteric lymph nodes, an increased number of inflammatory cells also in the mesenteric lymph nodes. This is this image. And also an increased inflammation, which was very high in the in the visceral adipose tissue of these mice compared to the mice associated with the other microbiota. So again, this is what you found in the human alcoholic liver disease. So in conclusion, different susceptibility to the liver diseases are transmissible by gut microbiota transplant, so demonstrating a causal role for the microbiota in these pathologies. I didn't show it during my presentation, but we were able to identify some bacterial species or bacterial genera associated with a high and low susceptibility to these liver pathologies, but at this stage more work are needed to decipher if these bacteria can trigger or protect from this pathology. And finally, gut microbiota transplant in germ-free mice could be used to demonstrate the contribution of the microbiota in other pathologies. And we believe that this study, the study of gut microbiota, so may lead to the identification of patients at high risk of liver diseases, of the severe stage of the liver diseases, and that targeting the gut microbiome may be in the future useful for the prevention of the treatment of the liver diseases. Finally, I want to thank the people from my group, and also Tiffaine Leroy and Martha Liopis, who are no more in the lab, but who did a large part of the studies I've shown. Our people from our germ-free facility, our collaborators, and our funding support for these studies. And thank you for your attention.